Gewoon, is nog 1 minuutje?
Ja.
Oké, ik ga gewoon beginnen. En dan lopen er nog mensen binnen en dan...
Ja.
Maar dat is denk ik helemaal oké. Go for it. Succes.
Ik ben helemaal van nu nog eens met je na te denken.
Hi, good morning everyone. Thanks for being here after such an amazing club night. First of all, I would like to thank the organizers of OHBM to invite us to present here today. And today I will talk about our consortium, the Leiden Consortium on Individual Development. And I think it's very unique that all of us are working on this same project in Leiden. So we're very excited to tell you about our work. So if you think of it, it has not been so long since we have been able to scan human brains non-invasively. And since we started to do that, we have learned a lot. Only a few decades ago, we started scanning children. And we have learned over the course of time that it's very important, if you want to know more about development, to follow individuals over time. Rather than only cross-sectionally, to really learn about developmental processes and how they interact. And only recently there are a number of projects around the world out there who have high quality longitudinal data sets and measures of MRI about developing individuals. And I think this is the time to make large contributions to solve pressing societal questions. And that is what we can do with the Leiden Consortium on Individual Development. As with this unique study design, we have collected high quality longitudinal MRI data and a very rich set of multi-model, multi-informant data of these individuals and their families. So I think these are very exciting times to work in the field of neuroscience and developmental neuroscience. So my name is Lara Wierenga, and I'm an assistant professor at Leiden University. And my co-presenters, Michele Achterberg, also is an assistant professor working at Erasmus University, Line van Drunen, and Simone Dobbelaar, both PhD students at the project working at Leiden University and Erasmus University. So this session is a little bit different because I will give a general introduction first about the whole project, so we're all on the same page, we know what it's about. And then we will all present our talks, and it are 4 pre-registered studies. And it's really exciting because we will show some preliminary results. And I would like to ask you to post your questions online or come forward here, and we will have room for questions after all of the talks and room for discussion. So the aim of Leiden SIT is to understand why some children thrive and others do not. And the main outcome measures that we're interested in is how social competence and behavior control develop, as these measures are important predictors of healthy individuals that make important scientific contribution to society. And therefore, with this project, we would like to understand the child characteristics as well as environmental factors that influence these outcome measures. So what we know is that these interact over the course of development, so think about gene-environmental interactions. But what is less known is how some individuals may be more susceptible to these environmental interactions than others. And the unique study design of Leiden SIT allows us to look at all 3 of them in one study. So all the different measures that we included are multi-informant, multi-method, multi-index, and having all of this information on the child and its environment makes it a very rich and unique data set. So we have set up a very innovative research design that optimally allows us to assess all of these interesting developmental questions. So we included 500 twin pairs that were followed 6 years annually with annual assessment. And what's exciting about this study's design is that we didn't include just one age cohort per two. So we started with the first age cohort already at 3 years old, and this other one at 7 years old. As you can see, there are 2 years of age range that are overlapping, and this allows us to assess the full age range from 3 to 14 years of age. We have very high quality MRI data, including T1 scans, TGI, functional imaging, and we have that both in a cross-sectional sample of 7 to 9 year olds and also longitudinally assessed biannually. And the early childhood cohort also contains brain measures of EEG, and we also have house visits and we have lab visits. Furthermore, this cohort includes an intervention, the video feedback intervention to promote positive parenting and to better understand why not all children are equally responsive to their social environment. We also had a natural intervention, like most of you have, by collecting data. So we were also in full lockdown a number of times in the Netherlands, and we had to be creative to continue the data collection. It happened exactly during one of the data collection waves. And we did so by partly online assessments, and we also replaced part of the house visits by online assessments. And in addition, we were able to keep on scanning, and we were very excited to do that with limited delays, and that was really by the hard work of our team and the PhD students. And of course, this is not where we stop. We are aiming to continue to follow up these individuals for another 10 years. So we're currently applying for funding to be able to do so. So the design of this study allows us to test a variety of different kind of research questions about development, because we have longitudinal data, about sensitive windows, we have the twins, so we can do genetic modeling, we have susceptibility markers, so we can look at differential susceptibility, we have overlap in the age cohort, so it also allows us to do replication within our own cohort. And I think this is very important today in this field. And with the intervention, we also have possibility to look at causal effects. So we don't only want to make a scientific contribution, but we also value open science. And for us, this is very important for a number of reasons. And I think this is really our core values. So we think that by being open and transparent, we can improve scientific quality. It will save limited resources and time, which is very important at this moment. We foster reproducibility and replication, and most of all, we also want to reduce participant burden, because we're so happy that they were able to come into the lab and keep participating in our work for all of these 6 years. So in our open science missions, we want to be transparent, we want to be findable, and we want to be accessible. And these 3 values is what we try to implement in all of our open scientific outcomes and products. So therefore, I'm very excited to show you that we have recently released a new website. Please check it out. It's very beautiful, and there are a lot of interesting open science products from our project on it, called leidensit.com. If you need more information, if you think like, oh, we would like to know more about your protocols, we are very open to share everything, so please reach out. And I would like to highlight that this whole project is a huge team effort. So we have been working on this for the last 8 years. First of all, our PI, Evelien Kronen, is really the true founder of this cohort. She's the brilliant mind behind it. We have many PhD students, many research assistants, and over 140 students helped in this data collection. So we are all very excited today to work in the field of developmental neuroscientists, and we already have a number of wonderful publications from the study. Please look them up. You can find them on our website. But and I would like to highlight that today we are going to talk about. We're going to listen to 4 pre-registered studies, and the results are preliminary. So. So you can post your questions online and we will answer the questions after each talk. So our first talk is from Michelle Achterberg, who was involved in the study from the start. So she wrote her dissertation that received multiple awards. And she's now a junior PI and assistant professor at Erasmus University. The floor is yours.
Thank you, Lara, for the really nice introduction of the LightAssist project. And thank you guys for being here at this early hour, the day after the quite epic club night. So I'm really excited to show some results that came out of the LightAssist project. So Lara already mentioned, my name is Michelle Achterberg. I'm an assistant professor at Erasmus University Rotterdam in the Netherlands, and today I will present on individual differences in developmental trajectories of social emotion regulation. So the leading question that drives me in my research is, why are some children more sensitive to social evaluations than others? And I try to answer this question by using a developmental neuroimaging perspective, so mostly focusing on functional brain mechanisms. And as Lara introduced, the ELSE project focuses on early and mid-childhood, and my research line on social emotion regulation, it mostly focuses on the middle childhood cohort. So childhood, and specifically middle and late childhood, it's the phase in life when the first long-lasting friendships, they really emerge. So dealing with social situations, such as social feedback or social rejection, it's really an important prerequisite for developing, but also for maintaining these social relations. So some children, they just shrug after a child rejects them on the playground. They keep their head high and they just move on. However, other children, they can get really frustrated and use reactive aggression after they receive negative feedback. So what determines how a child reacts, and how come that some 9-year-olds behave more adult-like than others? And we think that this might be due to individual differences in developmental trajectories of social emotion regulation. So that is, children, they substantially vary in the quantity and the quality of the friendships they have and the extent to which they feel supported by their friends. And that affects both the behavioral and neural responses to social interactions. So by taking into account these individual differences in development, we can address the fact that the children and their brains develop in meaningfully different ways. And this is particularly important when we look at behavioral and neural responses to social interactions. So to experimentally examine behavioral and neural responses to social interactions, we developed the social network aggression task. So prior to the lab visit, children filled in a personal profile with questions such as, what is your biggest wish? And during the scan, they saw pictures of unknown peers with social feedback towards the profile. And this could either be positive with a green thumb up, negative with a red thumb down, or neutral, which is just a gray circle. So after the feedback event, children get the opportunity to react towards the peer that judged them by blasting a really nasty and loud noise. Good morning. So children can choose themselves whether they want to have a short and soft sound or really a long and nasty sound. So in my research, I specifically focus on the neural activation during the feedback processing and how this relates to behavior aggression. So in the past 7 years, we've done several studies using this paradigm, and both in children as well as in adults. And behaviorally, we find strong effects in different samples that all show that negative feedback leads to the most aggression. So the cross-sectional studies, they show that older participants differentiate more between conditions. So the difference between conditions, how aggressive they are, it becomes more pronounced. And of course, as a neuroscientist, I am particularly interested in the neuromechanisms underlying this behavior and how these behavioral responses relate to brain development and brain maturation. And for this, this can study using the LIDA-SID project. So as Lara already explained, the study includes 2 cohorts, and I focused on mostly the middle childhood cohort, which included over 500 kids at the first measurement. So all participants in the study are same-sex twins, and approximately half of them are monozygotic. So this allows us also to perform behavioral genetic modeling on neuroactivation. In the scope of time, I haven't included that in this presentation, but if you're interested, then please feel free to ask me about this. So for my PhD research, I focused on the first 2 MRI waves. And in my current project, I look at development across 3 MRI waves. And currently, Lara and I are planning to include an additional MRI wave, the 3rd MRI wave, which we will collect around next summer when the children are around 15 years old. So using this data set, what we found, and what also other studies found that used the social network aggression task, we experimentally showed that there's a neural network that is sensitive for general social salience, so irrespective of its salience. So both positive and negative social feedback resulted in increased neuroactivation in the bilateral anterior insula. And interesting, variation in the anterior insula has been related to variation in aggression regulation. So we previously found that children with increased activation in the insula showed more aggression after social rejection. So interestingly, Dave Chester and colleagues, they found a similar association in adults, but only in adults with low executive control and not in adults with high executive control. So possibly the association between insular activation and behavior aggression is stronger in childhood than in adolescence, as executive control function increases across development. So the current study includes longitudinal measures across childhood and emerging adolescence, so we can actually test for developmental changes in brain behavior associations. So a second brain region that is often implicated in social behavior is the medial prefrontal cortex. So when conducting whole brain analysis on social rejection, previous studies often fail to find significant neuroactivation after negative social feedback. So in studies with a larger sample and strong statistical power, we actually do report strong activation in the medial prefrontal cortex after social rejection. So possibly these effects might only be detectable using these large samples because of the many individual differences in development of medial prefrontal cortex. So for example, in our test and replication design, we failed to find significant effects in separate samples, but if we combined them exploratively, we did find significant activation in the medial prefrontal cortex. And using the complete LIDA-SID sample that were included over 350 MRI scans at the first waves, we also reported strong activation in the medial prefrontal cortex, specifically after social rejection. So possibly these effects might only be detectable using larger samples because of the many individual differences. So both the insula and the medial prefrontal cortex seem to be related to processing of negative social feedback. And there are some associations found to behavior or to aggressive responses after negative feedback, but these associations are not yet clear. And in the next upcoming years, we want to really extend our work on these brain behavior associations and how they change across development. So one brain behavior association that has been consistently found using the social network aggression task is the negative association between dorsolateral prefrontal cortex activation and negative feedback and noise blastation. So if we look at the whole brain and we see which regions in the brain become activated, if we look at aggressive responsive after social rejection, we actually only see neuroactivation in the dorsolateral prefrontal cortex. So here plotted on the y-axis, you see the level of aggression, and on the x-axis, the dorsolateral prefrontal cortex activation during negative versus neutral feedback processing. And more dorsolateral prefrontal cortex activation is related to less aggression, so more aggression regulation. So interestingly, if we look at development from 7 and 8-year-olds to 9 to 11-year-olds, so the first 2 MRI waves in our sample, we see that children that show the largest increase in dorsolateral prefrontal cortex activation across time were also the ones that show the largest decrease in behavior aggression. So again, here on the y-axis you see aggression, but then the difference score across time, and on the x-axis you see the dorsolateral prefrontal cortex. So stronger DLPFC development was related to larger decreases in aggression, so stronger increases in social emotion regulation. And this shows that it's not just something that you can or cannot do, but it's really something that develops, and specifically during middle and late childhood. So our prior work using the social network aggression task showed 3 important brain regions that are related to processing social feedback, so the insula, the dorsolateral prefrontal cortex, and the medial prefrontal cortex. However, to date, the analysis, also my analysis, they have focused mostly on between individual group level comparisons using aggregated scores. But by taking a within individual approach, we can actually move beyond the aggregated mean scores and provide more insights into these individual differences and individual differentiation in development. So in my current project, we will take advantage of the within individual experimental design to estimate within individual neural and behavioral responses to social rejection using a multi-level model approach. And if you're interested in all the details of this study, please also check out my pre-registration on OSF. So by moving beyond the simple averaging scores, we can actually take more advantage of the rich experimental data that we have in this design. So on a behavioral level, the social network aggression task has a 4-level structure with repeated trials nested within measurement waves, which are nested within individuals, and because of the twin design, these individuals are nested within families. And using a multi-level model, we can estimate individual responses to social rejection at the trial level, but allow these changes in responses across measurement waves, describe development for individuals at the individual level, and take the non-independence of the measures due to twinning into account. So we can then estimate the B0, which represents the random intercepts, and B1, which represents a random slope. So these are interpreted as an individual response to social rejection. And both of these can vary across measurement waves. And across individuals and across families. So that's the subscribes I, F, and W. So at the second level, level 2, we can then estimate the development of individual responses to social rejection across age. And here the gammas reflect the growth components. And here we include linear and quadratic age components. So on a neural level, we actually have a 3-level model because the first level are the contrast values that we have from the contrast negative versus neutral. And then using that contrast, we can estimate the B0 and the B1, so the random intercepts and the slopes, and take that into account across waves and actually have the same model as we have for behavior. So I haven't completely run all the analyses, so I can't really show the brain behavior associations across time yet, but I do want to show you some preliminary results. And here I've plotted all the individual behavioral data of the social network aggression task. So on the y-axis, you see the noise blast that we just heard in milliseconds, and on the x-axis, I plotted age. So each dot in this graph is one measurement, and each small line is one individual. And the thick lines, they represent the group average. So these are the fixed effects. And as you can see across development, the noise blast towards neutral feedback, it gradually decreases. So children become less aggressive. However, for noise blast after negative feedback, we first see an increase and a later decrease around the start of puberty. However, what you can also see from this plot is that the individual data shows a lot of variation, again highlighting the importance of taking into account also the random effects or the individual differences. And of course, this is OHBM, so I guess you also like some preliminary brain data. And here I've plotted the dorsolateral prefrontal cortex activation for the contrast negative versus neutral feedback. On the y-axis, you see the parameter estimates of the DLPFC for this contrast, and on the x-axis, you see the different MRI waves. So as you can see here, there's only an effect during late childhood on a group level. So the fixed effects, there's significant differences in dorsolateral prefrontal cortex activation after negative and neutral feedback. And interestingly, this is also the phase where we saw the biggest differences in behavioral responses to social feedback. So something is clearly happening here around late childhood. So preliminary visualization of the data shows that both on a behavioral level as well as in the dorsolateral prefrontal cortex, there's an increase in late childhood followed by a slight decrease in early adolescence. And in our next step, we will examine this nonlinear pattern statistically and also conduct a brain behavior association, so to really test how these two trajectories align with each other. And we will then associate the random slopes of behavior to the random slopes of neural development. We also want to examine whether the individual differences in development, so these random slopes, are predictive for future social well-being, which we examined using questionnaires after 1 year after the third MRI wave. So if you want to stay tuned, please do follow the pre-registration or look me up at Twitter. So what I hope to show you is that there are strong development in social emotional regulation during childhood and emerging adolescence, and activation in the dorsolateral prefrontal cortex has repeatedly shown to be related to the level of aggression regulation following social rejection. And our preliminary longitudinal analysis, they already show that there's an interesting development going on during the late childhood phase prior to the start of puberty, around this age range, there's not just the start of puberty, there are also important environmental influences, such as the transition to high school, for example. And in the upcoming weeks, I will test how these individual differences in brain and behavior relate to each other. Hopefully, by moving beyond just a descriptive analysis, really towards these multimodal predictive analysis, we can use our longitudinal data across childhood to predict adolescent social well-being. And this way, we can really unravel which children are prone to thrive and which individuals might actually benefit from some more social and emotional support across this transition to adulthood. So with this, I want to thank you. I want to thank my co-authors on this project, Evelien Kroone, Simone Dobelaar, Stefan Heunes, and Jeroen Mulder. Also, I really want to thank the whole Leiden SIT team for all their hard work, and of course also my department at the Erasmus University Rotterdam. Thank you.
I would like to give the floor now to Simone Dobelaar. She's a PhD student working on Leiden SIT and focusing on the interplay between pro-social behavior and social emotion regulation. And her replication approach is very innovative, so I think you will be impressed.
Thank you, Lara. And good morning, everyone. Thank you all for being here. I'm very happy to present my work today on differential susceptibility effects on the development of social behavioral control. So my name is Simone Dobelaar. I'm a PhD candidate at Leiden University and Erasmus University Rotterdam, and I work also on the Leiden SIT project. And within my PhD, I focus on the social development from middle childhood to early adolescence, which spans approximately 7 to 13 years of age. And this is an important developmental period because it's marked by an increase in social experiences and social evaluations, so for example at school and among friends. And positive peer evaluations can result in pro-social behaviors, such as helping or sharing. But negative peer evaluations can also result in aggression or retaliatory behaviors. And such aggressive responses can then kind of lead to a negative spiral of even more peer rejection. Thus, it's a crucial period to learn to regulate and control your responses. I also call this social behavioral control. And within my talk, I will focus on 3 aspects of social behavioral control. So first, I will show a test replication design that we did to study neural activation and mechanisms related to social behavioral control in childhood. Second, we tested how social behavioral control develops between 7 and 13 years of age, and we did this both cross-sectionally and longitudinally. And third, we studied effects of the social environment on this development, and more specifically of parents. And we test for which children these effects might be most pronounced. Okay, so first I'll focus on neural correlates and age effects in childhood. Just like Michelle, I also used the social network regression task to measure social behavioral control. So in this task, quick recap, participants receive positive, neutral, or negative peer feedback on a personal profile, and subsequently they're instructed to send a loud noise blast towards the peer who gave the feedback. And they can decide the duration of the noise blast themselves, but they were instructed to always send the noise blast. So whereas Michelle's studies focus on this feedback processing event, I focus on the noise blast event to really study social behavioral control. And the design of the Leiden-Sit study allowed us to test our questions in 2 samples. So an independent test sample in the middle childhood cohort and another replication sample in the early childhood cohort. And both samples were 7 to 9 years old. Okay, so our fMRI results showed effects of feedback condition in both samples on activation in the dorsolateral prefrontal cortex during these responses to social feedback. And what we saw was that DLPFC activation was highest following positive feedback and lowest following negative feedback. And Michelle just explained that children also had the shortest noise blast following positive feedback, which could indicate inhibitory control processes. So we tested whether this was indeed the case by looking at the brain behavior correlation. And indeed, we found that activation in the dorsolateral prefrontal cortex was associated with social behavioral control. So more activation during this noise blast was associated with shorter noise blasts or more social behavioral control. So whereas Michelle found a relation between activation in this region during social feedback processing and behavior, which could possibly indicate emotion regulation, these results additionally show that it might also be an important mechanism for inhibition or behavioral control in a social context. We also found age effects in this relatively small age range of 7 to 9 years old. And what we saw was that behaviorally, older children showed less aggression or more social behavioral control following positive feedback. In other words, older children showed more differentiation in their behavioral responses to social feedback. And we actually found a similar pattern for activation in the DLPFC. So again, older children showed more activation during responses, but only to positive feedback. But to truly investigate development, it's important to look at it longitudinally, which is what we did in our next study. We again used the data of the Leiden-Sit study, and we studied this in the 3 MRI waves of the middle childhood cohort. And this spans the period between 7 and 13 years of age. So in this figure, you see what we just saw. So between 7 and 9 years of age, there was a rapid decrease in noise blast duration following positive feedback. But then from 9 years onwards, we actually see a rapid decrease in noise blast duration following negative feedback. Thus, there seem to be differential longitudinal trajectories for responses to positive and negative feedback, where social behavioral control related to positive feedback seems to develop a little earlier in childhood. For DLPFC activation during the noise blast, we saw a rapid increase in DLPFC activation during responses to positive feedback, which I just showed as well. But then from 9 years onwards, the DLPFC activation for both types of feedback seemed to slightly increase, but also seemed to follow a similar shape. Okay. Our final aim was to test for effects of the social environment on this development of social behavioral control. And more specifically, we investigated the role of parents. Parents are one of the most important socialization agents in childhood, so their behavior might affect the development of social behavioral control later in adolescence. We focus on parental sensitivity, which is the ability to notice, correctly interpret, and adequately respond to signals of the child. This has previously been associated with several positive developmental outcomes, such as increased prosocial behaviors or decreased externalizing problems. However, not all children might be equally responsive to changes in their social environment, so to changes in their parents' behavior. And the differential susceptibility model proposes that some children are more susceptible to their environment for better and for worse. So that means that they might be more vulnerable in deprived environments, but thrive in positive environments. And in our study, we want to test the hypothesis that temperament, which is a behavioral marker for environmental sensitivity, works as a differential susceptibility marker on the relation between parental sensitivity and social behavioral control. We hypothesized that children with a more reactive temperament or high environmental sensitivity might show less behavioral control in an environment of low parental sensitivity, but more behavioral control in an environment of high parental sensitivity. So if you're interested, you can read more in the pre-registration, which I added here. But for now, I'd like to show you some very preliminary results. So first, we tested whether the development of parental sensitivity in childhood was associated with social behavioral control in early adolescence. However, when we modeled the linear development, we did not find differences in linear slope between persons. So all participants showed a similar linear developmental trajectory. However, as you also can see in this figure, there were differences in the intercept, so the level of parental sensitivity at 7 to 9 years. So we tested whether the intercept of parental sensitivity was associated with social behavioral control in early adolescence. However, our results did not reveal a relation between parental sensitivity, the intercept of parental sensitivity, and social behavioral control in adolescence, so both not on a behavioral level with noise restoration, but also not on a neural level when we used DLPFC activation. However, we also did some exploratory bivariate growth curve models, and here we did find a relation between the intercept of parental sensitivity and the intercept of social behavioral control. So children of parents with higher parental sensitivity scores showed shorter noise blasts or more social behavioral control following positive feedback. Thus, these preliminary results suggest that parental sensitivity is related to social behavioral control in childhood, but not predictive of scores later in adolescence. This might be explained by increased influences of other social environmental factors, such as the increased influence of peers and friends. So, taken together, our application study revealed a robust neural basis for social behavioral control in the dorsolateral prefrontal cortex in childhood. Second, we found differential longitudinal trajectories for responses to positive and negative feedback, where social behavioral control related to positive feedback seems to develop somewhat earlier in childhood. And finally, we found no relation between parental sensitivity in childhood and social behavioral control in adolescence, but we did find an association in childhood. So our next step will be to test for differential susceptibility effects by temperament. And ultimately, I hope that these findings might help us understand why some children are more prone to aggression than other children, and also which children might benefit most from positive environments in order to thrive throughout their development. And with that, I'd like to thank my co-authors. I'd like to thank the Leiden SID research team, all participating families, of course, and thank you for your attention.
Thank you so much, Simona. That was great. So I would like to move on to Lina van Drunge. She's a PhD student, and she has developed a number of different tasks that she implemented and added to the cohort. And today she will talk about her work on sensitive periods, and she addressed this in a very innovative way. So go ahead.
Thank you for this introduction. Welcome everyone. Yes, today I want to tell you a bit more about sensitive windows, in which we used musical performance as a model of brain plasticity. And we used a longitudinal twin study on sensory motor synchronization and brain plasticity. My name is Lina van Drunen. I'm a PhD student at the Leiden and Erasmus University of the Netherlands. And I will present the results that I've been working on together with my fantastic co-author team, displayed on the left. So the human brain goes through a period of dynamic structural changes. And prior work already revealed that cortical and subcortical regions show rapid gray matter decreases between childhood and early adulthood and adulthood. Yet it is currently unknown whether these developmental patterns reflected in attenuated or accelerated declines are indicative of a sensitive window, where experiences would have heightened effects on brain developmental patterns. Possibly, musical capability can be an enriched environmental factor influencing brain developmental patterns, specifically during a sensitive developmental period. So there are many individual differences in musical performance, so only a few become musical experts. For example, the Dave Brubeck Quartet. I don't know if you know this band, but they're specifically very good in their great timing. And let me give you an example how this sounds. Well, they became experts through intensive training in combination with certain genetic predispositions. For example, 50% of the variance of rhythmic ability can be influenced by genetic factors. And one of the key elements of music is beat perception and prediction. Without this, music really sounds off. So it's a very important fundamental part of music. Additionally, age of onset of musical training in childhood can also be an important source of individual variability, where early training can contribute to brain development. So therefore, studying how musical performance relates to brain development offers us a unique opportunity to investigate the genetic and environmental effects related to this brain plasticity. A fundamental component of musical capability is sensory motor synchronization, and it can be measured with finger tapping paradigms. Sensory motor synchronization is defined as the ability to synchronize movements to auditory rhythms of an underlying beat. Well, this skill depends on several brain networks, including regions in the motor and the affective network, such as, for example, regions in the frontal area, which are mostly involved in motor preparation and execution, regions in the basal ganglia, so more subcortical, those are involved for memory formation, but also emotional processes that are involved in musical performance. The cerebellum, of course, for the fine motor learning, the temporal lobe, which is very important for beat prediction and perception. So musical training, specifically during a window of enhanced developmental plasticity, may be an important contributor to individual differences in structural brain development. So therefore, we assessed two aims. Our first aim is whether we could investigate the individual differences in structural brain developmental patterns in the motor and the affective network in 15 different regions of interest, and whether they are predictive of sensory motor synchronization performance in 7 to 14 years old. Second, we will test whether the relation between brain developmental patterns and sensory motor synchronization performance are driven by genetic or environmental factors, and we used a bivariate genetic modeling for this approach. So not much is known yet about the effect of enrichment of environment on brain plasticity in humans. However, most studies focus on deprivation on environment and brain plasticity. So prior work already reported that greater exposure to repetitive stress is associated with accelerated brain maturation. And they used socioeconomic status as example of enrichment or deprivation of environments. So based on these findings, we hypothesized that an enriched environment, such as musical training, can support brain plasticity during a sensitive window. Where we expected higher sensory motor synchronization performance is associated with attenuated brain maturation, reflected in more gradual slopes of cortical thickness change than children with lower sensory motor synchronization performance. So our twin sample included 3 waves of brain structure data, starting with 7 to 9 years old and ended up with 11 to 13 years old, 14 years old actually. And at wave 3, we collected sensory motor synchronization performance, and we collected data of a questionnaire, and this involved musical experiences across their lifetime. So for example, we know that 36% plays an instrument, 23% practice between 1 and 5 years, and 4% practice already more than 5 years. 6% started playing before the age of 7, 26% after the age of 7, and 64% did not play an instrument yet. So children were instructed in this paradigm to tap on a percussion pad with their dominant index finger. The first part was out-phase tapping, where children were asked to tap alternately with the tempo that was produced by the metronome. The second part was music-cued in-phase tapping, in which participants were asked to tap along with 4 different songs, 2 less challenging songs and 2 more challenging songs. I'm gonna let you hear an example of a more challenging song, and you can try this paradigm yourself. If you want, you can bounce your head, you can tap on your finger, you can clap along, whatever you want. Bruises on both my knees for you, don't say thank you. So we operationalized sensory motor synchronization performance by computing how stable children were tapping, relative to the beat. So our behavior results already revealed that children perform best in stability tapping in the out-phase tapping conditions and in a simple song condition, compared to the more challenging song conditions depicted on the right of the figure. And we use these 5 conditions as outcome variables of sensory motor synchronization performance in our neural growth modeling, which I will explain in the next slide. As an exploratory analysis, results revealed that children in the high socioeconomic status group perform better in stability tapping compared to children in the middle socioeconomic status group. And this effect was explained by the total amount of time played on an instrument. So children in the high socioeconomic status group played longer on an instrument compared to children in the middle SES group. And possibly these children have more access to resources. Our neurolatent growth model results revealed that cortical thickness decreased between 7 to 14 years old, which is in line with prior studies. But furthermore, volume in subcortical regions increased in this period. But we want to emphasize with these figures that the individual trajectories differ if you look closely to all the individual lines in the graphs. And we were interested mainly in whether these individual trajectories were predictive of sensory motor synchronization performance. In total, 5 out of 15 brain regions of interest were associated with sensory motor synchronization performance. And here I will present 2 examples of 2 regions of interest that were associated with sensory motor synchronization. In line with our hypotheses displayed on the left, we observed that indeed attenuated or delayed decline of cortical thickness in 2 frontal brain regions was associated with better sensory motor synchronization performance. The x-axis represents the slopes and the y-axis represents stability tapping, with 1 defined as perfect stability tapping. Well, as you can see, a less negative slope was related to higher sensory motor synchronization scores. As a sensitivity check, we explored whether socioeconomic status explained these neural results. However, this was not the case in all our regions of interest. For our second aim, bivariate genetic modeling revealed that for these aforementioned brain regions on the previous slide, approximately 13% of the covariance in heritability estimates between brain developmental slopes and sensory motor synchronization performance was best explained by shared environmental factors depicted in yellow. And we found similar results for the other frontal region, the pars orbitalis. So taken together, mainly environmental factors affect the variance of the overlap of brain development and sensory motor synchronization performance. So, I will hope that you will take away from this talk that sensory motor synchronization performance is related to structural brain developmental trajectories of the motor network, but also of the affective network. We observed that mainly attenuated brain development was correlated with higher sensory motor synchronization performance. And suggestively, there is indeed a sensitive window of brain development where enrichment of musical training affects brain developmental trajectories to a larger extent. And a substantial part of the relation between brain developmental slopes and sensory motor synchronization performance was driven by shared environmental factors and unique environmental factors. And possibly musical practice, which is an environmental factor, has a larger influence than musical talent, which is the genetic factor, on the individual differences on brain development. Thank you very much for listening. I want to thank the team again that have been working with me together on this project. If you're interested, you can read my pre-registration using this QR code. And furthermore, if you have any questions, I'm happy to answer these. Thank you very much.
Thank you so much, Lina. I will finish this today's session with a talk about sex differences in variability, and I look at that to better understand sex differences in vulnerability to mental health disorders. So there has been more focus in neuroscience to study sex differences in the brain, yet I still think we have a long way to go, as sex differences are often overly simplified, and this also has harmful consequences. And we are just starting to understand how all these different sex differences interact over the course of development. So we have the genetics, we have social-cultural influences and hormones, and now in most studies we start to look at these specific influences on the brain, but of course they interact over time. And today I would like to add some extra complex layer to studying sex difference by moving beyond mean differences. So I would like to note and make this disclaimer that this presentation is about biological sex, so having either two X chromosomes or an X and a Y chromosome, and not about gender identity or sexual orientation, which are probably have a different developmental pattern or influenced by different processes. So why would it even be interesting to study sex differences at all? So it is very striking that a number of psychiatric disorders show sex differences, show differences in their prevalence, with a general pattern of more externalizing disorders having a higher prevalence in males and more internalizing disorders having a higher prevalence in females. And what's striking about these externalizing disorders that they have often an earlier onset and very high heritability rates, up to 90%, for example, in autism spectrum disorder. And this may suggest that there is a biological mechanism involved in increased vulnerability to these disorders in males. So where does this heightened vulnerability come from? Maybe if we would better understand sex differences in the brain, we would be able to better understand and target these vulnerabilities. So neuroimaging research has focused on comparing male and female brains, and the single most consistent finding across studies is that the brain in males is on average 10 to 11% larger than in females. But if you look at the regional differences, it highly depends on the way this overall brain difference is accounted for. So here you see a sample of an image of a result of a large sample from the biobank data. And you see that if you control for this overall brain volume difference, the regional differences almost entirely disappear, or they sometimes even flip around. And there's very little consistency across studies. But more importantly, studying mean sex differences in the brain hasn't helped us much further in understanding how this relates to behavioral vulnerabilities. So with this approach, we still not do understand vulnerabilities in mental health well. So one reason for this may be that studies have mainly focused on studying group differences with the assumption that the brain is sexual dimorphic. But what we know from brain developmental studies is there is quite some variability in brain structure and development. And you see here data from the brain time studies that was collected in Leiden that no individual follows the exact average line. And what also stands out, there is large variability, larger within the group, so within males than in the average difference between the groups. And currently, we do not understand very well what these individual differences in each trajectories mean. So that was my motivation to look into variability in relation to sex differences. So one of the assumptions that underlies studying mean sex differences is that males have a Y chromosomes, females don't. This will cause differentiation in gonad development and also differences in hormone levels, which cause sexual dimorphic differences. But there's another mechanism at play in which genetics may affect body tissue. So females have 2 X chromosomes and males have only 1. And this would mean that the X chromosome will be expressed twice as much in females as in males. But the body has something developed to prevent this from happening, which is random silencing. So in each single cell in the female body, one of these X chromosomes is randomly silenced. And this has 2 different consequences. So first, any trait that is expressed by the X chromosome will be averaged within the female bodies because you have 2 copies of this gene. In a male body, you only have 1. So an extreme trait will be expressed throughout the body. This results in greater male than female variability of an X-linked or X-regulated trait. And the second consequence is what I refer to as the cat fur hypothesis, is that any trait expressed by the X chromosome will be more homogeneous within a single male. So this is also, in cat fur you see is also X-linked. So calico cats with mixed furs are always female. This is an oversimplified image, of course. There are also escape genes and homologous genes on the Y chromosome, so there are exceptions. And in 2013, an interesting study came out by Lara et al., and they showed that boys have greater variability in body size compared to girls at birth. And although the greater male variability was previously observed in personality and other behavioral measures, it was not really certain whether there was like a genetic component to it or whether that was more environmentally driven. And the fact that this was observed so early on in a biological measure before any social cultural effects could be in place suggests that there is indeed a biological mechanism involved. And I really want to highlight that it's important to look at both the extreme ends of the distribution. So much more for these personality and other behavioral measures, you see that this greater male variability is just at the lower side of the distribution, and the upper side of the distribution may disappear when, for example, with more equal opportunities for boys and girls. So what about sex difference in the brain? At this time, no one had looked at it yet. So I had a hypothesis that if there would be effects of X-linked genetics in the brain, then we would see greater male variability compared to female variability, that this would also be observed at both the upper and the lower end of the distribution, and also that we would see the cat fur hypothesis, so the brain of males would be more homogeneous than the female brain. So I first looked at this in the PINK study when I was working in San Diego in the lab of Jay Geet. We looked at a number of global brain measures in a sample of over 1,000 children, and we indeed saw that there was greater male variability in these brain regions, also showing great outliers at both the upper and lower extremities, and I think this is really important to test. then we wanted to know, well, yeah, if you want to look at variability, you need really large samples, so can we replicate this in a large sample? So we joined the Enigma lifespan group, and again, we saw like a striking, really strong effect of greater male variability in surface area and cortical thickness. And also at the bottom, you see a covariance matrix, which was stronger in males than in females for cortical thickness. And I also wanted to know whether you would see this in longitudinal, so developmental patterns as well. So I looked at the brain time cohort, which was collected in Leiden. And I also saw that if you look at the longitudinal trajectories, these are also more variable in males than compared to females. But okay, this is interesting and it seems like a consistent finding, but it doesn't help us much further if we cannot link it to behavioral differences. So how does this explain us to understand sex differences and vulnerability to develop these neurodevelopmental disorders? So this research paper came out by Nerdhoff et al., which indicated that there might actually be associations between mental health disorders at both extreme ends of the distribution if you look at functional, structural brain. So I hypothesized that maybe this will explain this greater male variability. So this is what I'm currently testing. And then also a beautiful paper by Sabi from the group from André Marquand came out, who looked at this from a different perspective. So they looked at autism spectrum disorder compared it to a normative group, and they saw exactly what I hypothesized, that these children with autism are more of the outliers at both the upper and lower extremes of the distribution. So I'm happy that they wanted to collaborate with me on this project. So we are currently testing this. So for the current study, and I can show you a few preliminary results, I used the Leiden SID study. I used both a cross-sectional sample and also are going to dive into the longitudinal measures. I have parent and child measures of ASD and ADHD symptoms and also cognitive measures from the children themselves. And I had the hypothesis that there would be, again, greater male than female variability within the brain. And we also have the twin sample, so this is really exciting. So I could dive into the covariance a little bit further, and I hypothesized that monozygotic boys who share the same X chromosome will have the most homogeneous brain, and that the dizygotic boys who might have different X chromosomes would show very less covariance, and that the girls actually would be in between. And furthermore, I'm very interested in linking this to behavior, and so I hypothesized that there will not be the linear association, but that the U-shaped association will be stronger if you look at the normative measures and link them to behavior. And what's really nice about using these normative models that you can also use smaller samples. Like ours, it's still large, but relatively small if you would like to look at variability. And on an individual level, you can find these outliers in the brain. So some preliminary results, I looked at the greater male variability, and you see here again that across the brain for cortical thickness and for cortical surface area at the top, you see that there is indeed greater male compared to female variance. And I also looked at the differences between all the correlation matrices within the twin pairs. And there, for example, for the subcortical matrix, I indeed find exactly this pattern where there's the strongest correlation between monozygotic boys and the weakest correlation between dizygotic boys. And I'm diving into these details to find like the networks that might give us some hints on where in the brain these X chromosome expressions are largest. So I would like to close off that even with these findings in sex differences, they are still small and there is a lot of other variability. So there's a lot of variability between individuals also adds up. So there is still more overlap than differences between the sexes. And it's really important to understand the full complexity of how these all interact across development. And I think it will help the field much further if we focus also on where sex is important, but also where it is not. So I would like to thank the whole Leiden SID team, and particularly my PhD student, Lina van Drunen, and my collaborators. And we are happy to answer your questions.
Thank you, Lara. Such a great presentation. So we already started answering some of the questions in the app, and we will also do so during the day. But if you want to stand up and ask your question, that's also good. And I can imagine you need some time to put in the questions for Lara or vote up other questions. Do you want to start?
Yeah, one of the high-rated question is, it's good to see that there are some developmental changes in the dorsolateral prefrontal cortex, but what are the biological mechanisms behind them, and how are you going to look into them? I think for you and Simone as well.
So I think that's a really, really nice question. So we do see these developmental changes in dorsolateral prefrontal cortex, and I think this is not solely due to puberty, because we actually see the biggest changes before the start of puberty. We do have to look into these, the start of puberty, because there are also individual differences therein. But we do have a lot of susceptibility markers as well, so hormones like testosterone, but also the DHEA. So we started measuring the testosterone at age 7, which might also be really interesting to look into that. And I think what's really innovative of this design is that they're all twins, so we can actually do the behavioral genetic modeling and look at whether this is genetic or biological, or whether this might actually also be developmental differences due to parenting or environmental effects. So I think this is really something we need to look into further, but we actually can, because we have all that data.
So you also answered now one of the other questions about the hormone levels. So yes, we do have hormone levels and pubertal measures as well. I see also another question that I can quickly answer. So will you also share the data? Yes, we are planning to do so. Very exciting. But we are currently now developing ways and tools like open science tools to do so in a very secure way, to keep respect of the privacy of the participants. And we are planning to release the data in May next week. Next year, sorry. So please reach out to us if you want more information on that. Another question for Lina. What about if the children knew the songs? How do you deal with that?
Yeah, you can hear me. Yes, the children were possibly familiar with the songs, and we specifically selected these songs because we wanted to be sure that the children were engaged with these songs. So we chose 4 songs in the top 100 of the Netherlands in the 2021, I think. But we also asked, after the paradigm, did you like the song? Were you engaged in the song? Were you do you want to dance to the song, for example, like were you reactive? And I think it helped us that they were more engaged instead of that they didn't know the songs, because otherwise they would be bored to tap along. So I don't think it's a problem, but we have the data so we can check whether it has an effect or not.
Thanks. Yeah. And a question for Simone. So how well do these in-scanner, non-naturalistic talks of aggression relate to the real-world behavioral aggression?
Yeah, that's a really a good point, actually. And we also do have a lot of questionnaires on, for example, aggression, but also behavioral control measures. And there we did find a relation with, for example, the Dutch reactive aggression skill. So in that sense, I think there is that is kind of a proxy of real-world aggression, but also sometimes you don't find it. And I think it's really interesting to also investigate this now in a later age range. So check whether that differs across age, and now that they enter into adolescence, whether it might be more predictive of the real world or not.
Yes, and for Lina, so you looked into the SMS performance in relation to SES, and that SES was also related to musical training. So did you look at the relationship between SMS performance and music experience?
Yeah, so if I'm understanding it correctly, do you mean other measures of musical experience? So we have a lot of measures of musical experience. We have, for example, whether the children were encouraged at home to listen to music, if they were going to concerts quite a lot, if there was music playing in their homes quite often or not. So I checked whether these were related to sensory-motor synchronization as well, but there was not as a strong effect as training. So we choose to include this in the model. However, we have so many measures, so it's very interesting to write a new paper about all the behavioral aspects of musical experience in these children. Yes.
So I also got a question on the sex differences. So how can you disentangle genetic and cultural influences in the prevalence of psychiatric disorders? And I think this is a very, very important question. And both processes are at play. We know that, for example, there's underdiagnosis of depression in males. There's like higher suicide rates in males. So therefore, I think also as a researcher doing science on sex differences in the brain, at the same time, it's really important to explain what our results mean to the public, because it can lead to misconceptions about how large these sex differences actually are. But it's very difficult to look at the environmental effects, so social cultural effects, because we all grow up in a society where there are differences. So even if you're a racial child, gender neutral, you have like kindergarten already, and school and environment, like if you walk in a toy store, there's already major differences. And most studies on looking at these gender-related effects look at children or people that don't have like the same gender identity as their biological sex. But there are also other problems there, maybe because their environment reacts differently on them. So it's really difficult to find out what the effects of cisgender are. And I'm gonna host a workshop in November in the Netherlands and get like a group of scientists and psychiatrists together to talk about how we can tackle this problem. And one of the ideas maybe is also to look at the cross-cultural effects. But yeah, this is really an open question, and I think very important to answer. Okay, let's see, are there any people who want to walk up to the microphone and ask a question that's not addressed yet?
So we don't mind if you're like a bit sore from the club night, we just listen very carefully.
Yeah, maybe this is also an important question to answer. So is there any screening for trauma or adverse childhood experience during this study? And how does this impact aggression?
So that's actually a really good question. We don't have data on early adversity for these kids. We did wanted to include a questionnaire on child trauma, but the Dutch Ethical Committee decided that that was too much. So we don't have that. We do have a lot of parenting measures within this study, and we also have questionnaires on stressful life events. But I do think it's super interesting to look at this specific task and this specific behavior in a sample with early adversity, and that's actually what we're going to do. So we're collaborating with Margaret Sheridan from Chapel Hill University, and she follows a sample that has early adversity when they were children, and now they are adolescents, and they will actually do also this task in MRI scanner, so we can actually check for the effects of early adversity on this social emotional regulation. I think there's a question there.
Yep. Oh, good morning. Thank you for speaking with us this morning. It's very interesting. One thing I was wondering is, as the cohorts age, especially as they enter adolescence, will you be looking at the effects of their personal relationships, like if they start dating and et cetera?
Yeah, so I think, thank you for this super interesting question. I think, so Lara and I were brainstorming a couple of months ago about how to, well, mostly how to minimize the study because it's super big and we don't have enough funding to do everything every year, but also like which measures are we currently missing, and there has been quite a few parental measures because I think parents are super important during childhood, also during adolescence. But now I think because those friendships are becoming more stable across late childhood and emerging adolescence, we can actually take that one into account. And we are also planning on to collaborate with Daphne van den Boongaard, who also works a lot on peer relations and romantic and intimate relations. So that is something that we can actually take into account, I think, starting the next wave, so next summer.
I think also an interesting question is, do they ever not give a noise blast?
Yeah, they actually were instructed to always send a noise blast, so they could decide the duration of the noise blast themselves, and if they didn't respond in time, then they got a message like you were too late. So I think that also explains my results, like that is really related to behavioral control, because you have to respond. So it's also kind of like you also have to be motivated to stop your response, which might be sometimes like even harder than just keep pushing that button. So yeah, so I think that is interesting, that we actually see that most in the positive feedback condition.
And a related question, did you also look into negative or like internalizing effects of aggression?
Yeah, so we did ask afterwards in the exit questions how they felt about the feedback, and then we actually, in all our samples, I think, we saw that they disliked the negative feedback the most, and they liked the positive feedback the most. Yeah.
Yeah, so I think for this paradigm, we really wanted to tap into those more reactive responses, because a lot of studies on social rejection actually focused on the more internalizing responses, so looking at depression or withdrawal. And I think this is really a study that looks at more the reactive responsive, but I do think that there is both, and I'm also interested in seeing whether children use either one of the other strategies, so either withdraw or be reactive, or whether this is context dependent, or whether this is dependent on mood, for example. So that would be super interesting to see. And I think, I hope that someday in the future we can also include some EMS studies, so on mood, but also on how you react to social reaction in the real world by asking questions, for example, through an app. And there we can actually also include whether they feel sad or whether they feel angry, and then make a bit more of that distinction. But for the task, they can only react aggressively. There's no possibility to withdraw.
Okay, I will close this session. Thank you all so much. Give them one more round of applause, please. And I would like to say again, if you have questions, if you want to collaborate with us, please reach out, phone us through our website. We will be happy to work together. Thank you.